What is microbial metabolism? Is all of an organism's chemical processes an emergent property that arise from interactions of molecules in the orderly environment of the cell. Metabolism is very important for the management of cellular material and energy resources. Two categories of metabolism: the catabolic and anabolic. Catabolism, destructive metabolism, the breakdown in the living organisms of more complex substances into simpler ones with the release of energy. Complex substance into simply ones release energy. Anabolism, constructive metabolism, the synthesis of living organisms of more complex substance from simpler ones, speeding energy. Simple substance into complex ones, speed energy. A part of energy released from oxidation of foodstuffs and from light gets stored in a molecule is known as adenosine triphosphate or ATP. It then transfers this energy through reaction that required energy. ATP functions as a carrier of energy in all living organisms including bacteria, fungi, plants, and animals. This is the ATP hydrolysis. ATP was discovered in 1929 by Carl Luhmann. It was proposed to be the main energy transferred molecule in the cell by Fritz Albert Lippmann in 1941. ATP is the nucleotide consisting of a purine base, a pentose sugar, and the 3-phosphate group. Adenine is attached to the 1-carbon atom and the phosphate group are attached at 5-carbon atom of ribose. ATP is the active form exists as a complex of ATP with Mg2+ or Mn2+ ions. ATP is a derivative of AMP to which two additional phosphate groups are attached through an anhydrous linkage. And those are particularly reactive, hence ATP is able to donate phosphate group to number of metabolic intermediates, thereby converting them to active forms. In AMP, where adenosine is attached to phosphate by an ester linkage, therefore less free active and termed a low energy band. The two pyridine nucleotide, the NED and the NEDP. Both of these pyridine nucleotides can undergo reversible oxidation and reductions. In catabolic reactions, oxidized pyridine nucleotides are the usual reactant. In the biosynthetic reactions, and reduced form is the usual reactant. Consequently, two kinds of pyridine nucleotides are required. Indeed, in cellular pool of NED, it's maintained largely in oxidized state, and the pool of NEDP is the maintained largely in the reduced states. Metabolic diversity in microbial world. First, green plants are photosynthetic. The second is animal required performed organic molecule for energy as well as building block. Autotrophs. The organism that driven carbon from CO2 plus. Heterotrophs, the organism using organic compounds as the carbon sources. Phototrophs are the organism using light as energy source. Chemotrophs include the organism carrying out chemical reactions to obtain the energy. Microorganisms also required a source of electrons for the reactions. Lithotrophs, microorganisms that driven the electrons from inorganic compounds like molecular hydrogen or reduced sulfur compounds. Organotrophs, in this organism, the electrons are donated by organic substrate. Organoheterotrophs. Press 1. Breakdown of the large complex biomolecules like polysaccharides, proteins, and lipids into their respective building blocks. Press 2. The building blocks are usually oxidized to a common intermediate acetal or CoA. Press 3. It consists of the citric acid cycle. Example, the oxidation of acetyl CoA to CO2, formation of NADH and FADE2, followed by electron transport and oxidative phosphorylations. Pathway is also known as EMP pathway. It is the most common pathway for glucose degradations for pyruvate, and it's found in animals, plants, and large number of microorganisms. This pathway is also by anaerobic as well aerobic organism. This process takes place in the cytoplasm of prokaryotes and eukaryotes. The pathway consists of 10 enzymes, catalyze reactions that begins with a glucose molecule. This reaction comprises 3 stages. First, conversions of glucose into fructose 1,6 or biophosphates. Second, splitting of fructose 1,6 biophosphate into 2,3 carbon fragments. 3. The formation of pyruvate along with ATP regenerations. The TCE cycle was first discovered by Eggleston and Krebs in animal tissues. It is also called as Krebs-citric cycle. The cycle is considered a central pathway of aerobic metabolism as it serves 2 purposes. Bioenergetic. The cycle carries out complex dehydrations of acetyl group and acetyl CoA to CO2 plus, resulting in release of energy ATP of GTP and reducing power of NADH and FADH2. Biosynthetic. It supplies precursors for several biosynthetic pathways of amino acids, pyridimidines, purines, and etc. The 3-carboxylic acid cycle. The cycle may be divided into 3 stages based on the size of its intermediates. The 3 stages are separated from one another by 2 decarboxylation reactions, in which carboxyl group are loss of CO2. The pyruvate dehydrogenase complex forms acetyl CoA through pyruvate oxidations.